Assalamualaikum warahmatullahi wabarakatuh. My name is Amir Ashraf. Today, I want to explain the experiment of restriction enzyme digest. In this simulation, I will be performing a restriction digest. I will use restriction enzymes to cut the DNA strands of two plasmids, producing DNA fragments. Because we know the size of our plasmids and where the enzymes will cut the DNA, we can predict the length of the fragments that will be produced. We can also confirm the length of these fragments or determine the length of fragments produced from an unknown plasmid construct using gel electrophoresis. These DNA fragments can then be joined to make recombinant plasmids. The materials that I will use is reagents, which is buffer 2.5xp solution, pKn R plasmid K solution, paraplasmid A solution, restriction enzymes RE, distilled water H2O, micropipetting equipment, P2 pipette, P20 pipette, P200 pipette, P1000 pipette, P2 tip box, P20 tip box, P200 tip box, and P1000 tip box. I will also use empty tubes such as A plus tube, A minus tube, K, K plus tube, K minus other equipment such as microcentrifuge, trash, floating tube, freezer, water bath, and also ice bucket. So, first step for restriction enzyme digest is set the temperature of the water bath. Select the water bath to set the temperature to 37 degrees Celsius. Next step is set the volume of the P20 micropipette. Select the P20 micropipette from the micropipette rack. Select the volume setting to set the volume to 4 microliter and then select Save Volume. Next, select the P20 tip box to open it. And then move the P20 micropipette onto P20 tip box to attach a tip. Close the P20 tip box. Step is draw up the reaction buffer into the micropipette. Move the restriction buffer 2.5xp tube to the empty tube rack and open it. And then select the micropipette and place it in the restriction buffer solution tube. Draw up the restriction buffer by selecting the plunger and holding it down until it reaches the first stop. Then close the restriction buffer. The next step is pipette the restriction buffer into the K plus tube. So select K plus tube, you open it. And then dispense the restriction buffer into the K plus tube by holding down the plunger until it reaches the second stop, then releasing it. Next, eject the tube and throw into the waste container. Next, close the K plus. So the next step is pipette the restriction buffer into the K minus, K plus, and A minus tubes. So follow the micropipetting procedure to dispense 4 microliter of this restriction buffer into the K minus. Eject the tip and screw into the waste container. Next, follow the micropipetting procedure to dispense 4 microliter of restriction buffer into the A plus tube. Then eject the tube, screw into the waste container. Next, follow the micropipetting procedure to dispense 4 microliter of restriction buffer into the A minus tube. Eject tube, screw into the. The next step is dispense the other solutions to the K plus, K minus, A plus, and A minus tube. Follow the micropipetting procedure to dispense 4 microliter of pKan R plus mid K from the K tube into K plus tube. Check the tip. Then follow the micropipetting procedure to dispense 4 microliter of pKan R plus mid K from the K tube into K minus. Eject tube, screw into the waste container. Next, follow the micropipetting procedure to dispense 4 microliter of para plus mid A from the A tube into the A plus tube. Check the tip. And then follow the micropipetting procedure to dispense 4 microliter of para plus A from the A tube into the A minus. Eject the tube, screw in twist. Next, change the volume of the P20 micropipette to 2 microliter. And then follow the micropipetting procedure to dispense 2 microliter of restriction enzymes from the RE tube into the K plus tube. Get the tube. Next, follow the micropipetting procedure to dispense 2 microliter of restriction enzymes from the RE tube into the A plus tube. Get the tip. Next, follow the micropipetting procedure to dispense 2 microliter of distilled water from the H2O tube into the A minus tube. negative, and then follow the micropipetting procedure to dispense 2 microliter of distilled water from the H2O tube into the K tube. Then eject. Next step is spin the solution tube. Select the microcentrifuge to open it, and then move the K plus, K minus, A plus, and A minus tubes to the microcentrifuge and balance them correctly by placing the tubes at equal distance from each other inside the microcentrifuge. Next, select the microcentrifuge to close it, and then set the microcentrifuge to spin for 10 seconds. Once the spin is complete, open the microcentrifuge by selecting it, and then move the solution tubes into the floating solution tube rack. Next, select the microcentrifuge to close it. Next step is incubate the solution tubes at 37 degrees Celsius. So select the water bath to open it, and then move the rack containing the K plus, K minus, A plus, and A minus tubes to the 37 degrees Celsius water bath, and then close the water bath. Next, set the timer by using the plus and minus buttons to 1 hour and press start. So when the time is up, remove the floating solution tube rack from the water bath by pressing the eject button. The last step is store the solution tubes in the freezer. Select the freezer to open it, and then move your rack containing samples K plus, K minus, A plus, and A minus onto the negative 20 degrees Celsius freezer, and then close the freezer to store your samples for later analysis.